Nature is a battlefield. Now, a group of animal rebels emerge, armed with a new and deadly chemical weapon, ready to paralyze, kill, and even take hostages.
Nature has designed the absolutely perfect bioweapon.
Can anything stand in the way of venom? In the baking heat of the Australian outback, the most venomous snake in the world, the taipan, stalks its prey. 700 times as toxic as a rattlesnake, the taipan's bite packs enough poison to kill more than 60 humans. As it enters a rat's burrow, it's armed and dangerous. In an instant, it strikes. In seconds, the rat begins convulsing, its internal organs under assault by a complex mix of toxins that ensures its death. Venom.
If you're a snake, having venom is a great asset because it means that you can grab an organism, inject it with venom, and then let it go, and you don't have to fight with it.
For hundreds of millions of years, the world's top predators have relied on speed, or their size and sheer strength, to overpower their victims. But venom shifts the balance of power.
When we think of natural selection and evolution, we think of animals chasing each other and needing to be fast or needing to be strong. But venom changes the rules a little bit. It means that the small can defeat the big. It means that the slow can beat the fast.
Inside a tiny drop of venom, there's a minefield of toxins, a sea of exotic chemicals, some bafflingly complex, others so strange, it's taken scientists years and millions of dollars to copy just one. Venom is such a potent and successful weapon, it has evolved at least 10 separate times within dramatically different animals. From the stinging cells of jellyfish, like the Portuguese man-o'-war, to the dripping fangs of the funnel web spider. There are venomous insects, venomous sharks, and even a venomous mammal, the platypus. So how did such a vicious weapon evolve? The answer may lie in a lab in Australia. Any investigation of venom must start with the animals that have killed more humans than any other: snakes. The scientist in charge is Brian Fry, the world expert on the evolution of snake venom.
It's a magnificent snake.
Collecting venom is laborious and dangerous.
All right.
For Fry, a day at the office can involve milking up to 50 different species of snakes. Every one of them a killer.
This is the Australian mulga snake. So this is a brown snake. What we've got here this time is a death adder. They're actually related to the cobras. So what we're doing is we're milking all of these snakes so that we can have the venom to study in the laboratory. You have to get the venom, obviously, from only one source. It's got a good little yield on him. That's something you see a lot of variation in snake venom, is the color of it.
Sometimes the process goes wrong.
Like I had one bite from one of these kinds of snakes where it bit, swung the fang, stuck it in, swung the other fang, stuck it in, swing, and just walked across my hand. I had a series of puncture wounds going across it. That was fun.
A dose of anti-venom saved Fry's life. But every year, 125,000 people die from snake bites. No anti-venom works for all snake bites because venom is so incredibly varied.
Venom is the ultimate in the movable feast, where not only do you get big variations from one species to another, but even within the geographical range of a single species, you can get quite pronounced differences in the venom composition, where they might have a different menu, you might say.
Fry has dedicated his career to working out why venom is so diverse and so deadly. Could its evolution be the answer? Fry already knew venom was a combination of deadly chemicals that worked in unison.
Think of it more of like basically a gang beating you to death with different shaped iron bars. It's a concerted, multi-dimensional attack on your body.
His quest to understand venom would take him 5 years. For Fry, just knowing it was complex wasn't enough. He wanted to know where all those chemicals came from and how they evolved.
It's a lot like sort of doing a molecular detective story. What we do is we break it down, we separate all the different components out into individual components, do a wide variety of tests and other analyses to find out what makes it work, what makes it kill, and how can we use it.
Fry first tested the effects of hundreds of different snake venoms, observing their terrifying impact on animal tissues. Some dissolved flesh like acid. Some made organs like the heart burst from the inside out. Others caused instant paralysis. He then used the latest technology, called liquid chromatography, to separate out the chemicals within each venom. On these graphs, each peak indicates a different killer toxin within just one venom.
So what we've done is we've separated this complex mixture into each of their individual components, where these have been collected into their own vial. We now have a fingerprint of this venom.
What these venom fingerprints revealed was startling. The chemicals in each snake venom had evolved from proteins, not just any proteins, but the universal building blocks used in almost every animal body to govern the most important physical bodily functions, from the circulation to digestion and even the chemistry of the brain. The twist was the proteins had evolved into new versions that could kill.
So the snake venom proteins don't come out of thin air. What they are is they're mutated forms of natural body proteins, often ones involved in very important physiological processes.
A snake's venom glands are biological warfare factories. When venom enters a victim, the killer proteins wreak havoc.
You can think of it as a smart bomb. It's built for the inside of the body that it's going to, and it's going to work there because that's where evolution has designed it to go.
So how does this mechanism work in practice with the most venomous snake of all, the taipan? To see the effects of its venom in action, Fry experiments on his own blood.
We've just taken a not small amount of my blood out, and we're going to use that. We're going to add taipan venom to one vial and leave the other vial alone, and then we'll be able to see the difference. So what I'm doing is adding the taipan venom to the blood. This is my blood. Without any venom, this is my blood on taipan venom. You can see what it's done to it. It's turned it into a solid clot. Now imagine this is my blood, but my blood actually inside of me. Obviously, this is not going to do good things. By making all those millions of tiny little blood clots, all your blood clotting factors are being used up. What this means is that you now can't clot, so you'll be bleeding out of your gums, your ears, your nose, everywhere else. It's not a nice way to go.
The taipan's venom evolved from proteins that formed clots in its own blood to heal wounds. Once injected, it reduces its victim's blood to a mixture of massive clots and a liquid that leaks from every orifice. When the taipan bites a human, venom surges into the body and peppers the bloodstream with millions of tiny blood clots. This triggers a cascade of deadly effects, shutting down the liver, kidneys, and lungs. All it takes is a fraction of a teaspoon. One mystery remains about the taipan. Out of the thousands of snake species on Earth, why is the taipan's venom so powerful? Fry thinks there's a reason for that.
So this is the Australian taipan, all of its legendary glory. Unlike any other Australian snake, they specialize on feeding on very dangerous prey items. Snakes normally don't try to go after these kind of prey, such as bandicoots and the native plague rat, because these are animals that could severely injure or even kill the snake.
In this footage, a plague rat can be seen taking a bite out of the taipan. That's why, over millions of years, the taipan had to evolve ever stronger venom in order to knock out its prey as fast as possible. Science had solved the riddle of how venomous snakes had engineered this remarkable weapon, but deeper mysteries still remained. When did venom first evolve? And where did it come from? Biologist Brian Fry had discovered how snakes had turned their bodies into bioweapons, engineering venoms of staggering diversity. And that diversity keeps evolving as snakes tailor their venom to suit their wide-ranging habitats, including even underwater.
The sea snakes are incredibly venomous snakes. And the reason for this is that in the aquatic environment, they don't have the luxury of tracking their prey. If the prey gets away, that's it. Someone else, like this little ray here, is going to make an easy meal out of it. So they need their incredibly toxic venom in order to make sure that they get their meal.
Sea snakes evolved from land snakes only 30 million years ago, and they're still refining their venom. They prove that snake venom is still adapting. But when did its evolution first begin? When Fry first set out to solve this mystery, it was assumed that venom was a recent innovation. It was all to do with snakes' extraordinary fangs. Fangs are hollow hypodermic needles through which venom flows under extreme pressure. This is how the fangs of a spitting cobra can propel its venom as far as 8 feet. They're so perfectly engineered to deliver venom that it was assumed fangs and venom had to have evolved together. But when Fry inspected the fangs of modern snakes, he found something intriguing. 3 major groups of snakes had evolved very different fangs.
You've got different lineages that have hollow front fangs, but of very different types, where the vipers, like the American rattlesnakes, swing on hinges and are very long. The cobras are short and fixed, while the stiletto snakes, they pop out sideways.
For Fry, this meant venom must have existed before the different types of fangs. Sure enough, when he and other snake experts across the world traced the evolution of venom back in time, they discovered the first snakes had no fangs, but they did have venom. For Fry, this suggested something strange. Maybe snake venom didn't start with snakes at all. Rather, they'd inherited their venom from an earlier ancestor. Snakes evolved from lizards 100 million years ago. Could they be the source? Fry decided to study them.
But they can be quite tenacious once they latch on. Now, better this than my hand.
Fry's theory went against common wisdom. Out of 5,000 lizard species, only 2 were known to be venomous, and they were considered evolutionary freaks. Fry decided to search for other venomous lizards. He zeroed in on the most ancient lizard species in the world, monitor lizards, such as the Komodo dragon. Komodos are fast ambush predators that can grow to the length of a car and weigh as much as a linebacker. The bites from their serrated teeth result in rampant infection that eventually kills their prey. Scientists had long thought this was caused by bacteria, 80 species of which are known to exist in the Komodo's saliva. But this explanation didn't satisfy Fry.
It never sat right to me about all this stuff about the toxic bacteria and Komodo dragons because, well, bacteria don't work that way. They don't cause rapid bleeding, muscle pain, and neurotoxicity within the first few hours unless it's some sort of escaped bioweapon or the Andromeda strain.
Fry wondered if there might be something other than bacteria triggering toxic effects in Komodo's prey. To find out, he would have to take a closer look. Using a brain scanner, he imaged the head of a Komodo dragon. It was then he found something that had been totally overlooked.
With the Komodo dragon, you can see a very large bulge running the length of the lower jaw. And with our magnetic resonance imaging, which is the same technology that you use to scan a brain to look for cancer, you can see the bulge there. Now, if you look in with the beauty of technology, you look within it, you can see a very complex gland arrangement. And that's actually a venom gland.
Fry had discovered a venom gland, similar to that of snakes. Might there be other venomous lizards? There was only one way for him to find out.
So to decide what was happening, we ended up going out and collecting as many of these things as we could.
Fry's team swabbed the mouths of hundreds of lizard species for saliva samples. He isolated their active molecules and tested them on animal tissues. The results were remarkable. The molecules broke down the animal's heart muscles and toxified their lungs, the same way as the venom of certain snakes. This wasn't bacteria. This was a slower-acting form of venom. Fry's team eventually found snake venom components in more than 1,500 lizard species. He had rewritten the evolutionary record.
We started looking at what was actually the chemical composition of the venoms and found that there was several components that were shared between the lizards and the snakes. And this was the final proof that they shared a common venomous ancestor.
It's now thought that snake venom, the snake's deadliest weapon, started its evolutionary journey in ancient lizards, millions of years before the snake itself first appeared. But these were not the first animals to evolve venom. In evolution, useful things tend to evolve more than just once. It's true of jaws, it's true of eyes, and it's true of venom.
Venom's appeared in several different groups of animals all over the world. It's appeared in fishes, in mammals, in insects, all independently. And when it shows up in a group, it stays because it gives them such an advantage over the animals with which they're competing.
A riddle remains, though, about venom. If venom gives such an advantage, why does it exist in only an elite few? Why don't all animals have it? After snakes, scorpions rank as the second deadliest group of animals in the world. They kill, on average, 5,000 people per year, injecting venom into their victims from their tail-borne stingers. Venom has helped scorpions to flourish in harsh desert habitats for more than 300 million years. But for all its advantages, could there be a disadvantage to having venom? That was the question Zia Nassani decided to explore, using one of the world's most toxic scorpions as his test subject.
The species that I work with is called Parabutis transvollicus. It's a butid scorpion, one of the largest scorpions out there.
Nassani studied their lifestyle intensively, their nocturnal hunting patterns, preferred desert habitat, and their use of venom. He found that a scorpion's venom gives it an enormous edge. But what happens when it has used up its supply? Nassani devised an experiment to compare one scorpion armed with a full gland of venom and the other completely drained of its venom. He used a miniature oxygen tank to measure their activity rates and found a startling difference. The milked scorpion used up to 50% more energy than a scorpion with a full supply of venom. Once having deployed its venom, a scorpion's body shows the same signs of extreme fatigue as a marathon runner after a race. Just as an athlete struggles to replenish protein and electrolytes, a depleted scorpion has to devote most of its energy toward remanufacturing its most critical asset.
Venom does have a trade-off, and that is it's not good for multiple uses. You have to use it and then make more. And every time you use it, you have to produce more with your body, and that has an energetic cost.
For the scorpion, there's another disadvantage to venom. After it's used, the animal is effectively defenseless for days.
When their venom is completely depleted, till day 4, they have to be very cautious in their foraging habits because they're susceptible to predation. Also, they have to limit themselves to capturing smaller prey items, so that leaves them at a competitive disadvantage.
These drawbacks may explain why venom is not more common in the animal kingdom. But there's a bigger reason why many other animals haven't evolved venom. Venom needs complex, sophisticated biological engineering to inject it.
In order to have a venom, you need to have a way of delivering it as well. And there's been a lot of different ways that this has been evolved, whether it be the fangs of the spider, the barbs of a stingray, the spines of a stonefish, the spurs of a platypus, et cetera, et cetera. Each one has their own unique way of delivering it.
And these customized pieces of hardware suck down even more biological energy, making venom not just a superweapon, but one of the most biologically expensive innovations that animals have ever evolved. Every venomous animal has a dilemma. It's armed with a deadly weapon that's a huge burden to maintain. Each has evolved a perfectly precise method to make venom work for it as efficiently as possible. None more so than the animal that turns its victims into zombies. An elite group of venomous insects, ounce for ounce, carries more chemical weaponry than any other animal. Each of them has evolved its own deadly delivery mechanism. Stings to pierce flesh, fangs to mangle prey. Insect venom is a world of elaborate microengineering. And one insect has taken that to the extreme, twisting its venom for a diabolical purpose that's like something out of science fiction, turning another animal into a zombie. These are the emerald jewel wasps. In this lab at the University of California, Riverside, researchers want to know how the wasp's chemical arsenal evolved to completely control her host.
So I first became fascinated with venoms because they represent a kind of chemical warfare.
Jewel wasps are parasitoids. They paralyze their prey so their offspring can live off them. After decades of research, scientists are beginning to understand the secrets of parasitoid venom. Females do battle with a host who is plentiful, widely available, and 6 times her size. The American cockroach. Researcher Chris Banks has devised an ingenious method to experiment with the wasp's venomous attack.
We're going to put a wasp and a cockroach into this area right here and let them interact. We've also included glass vials here, and these will serve as makeshift burrows for the wasp.
An unsuspecting host is first introduced to the arena. The roach is unaccustomed to threats from smaller animals. The stage for the battle is set, and the wasp is released. As soon as the jewel wasp senses a host, she will surprise her prey and use that brief moment to her advantage. She attacks. The wasp's first sting is brilliantly strategic, just enough to disarm the roach.
The first time it stings it into the central nervous system and causes a temporary paralysis of the front legs.
Venom hits the roach's nervous system in between its front legs, briefly paralyzing them.
This allows the wasps to do a second, much more precise sting into the brain.
The wasp twists its body around the roach so it can sting into its brain and inject the zombifying mixture. The roach's breathing slows. It makes no move to escape.
This brain sting causes a dramatic behavioral change in the cockroach, basically turning it into a zombie.
Wasp venom is precisely engineered to shut down signals carried by a key brain chemical called dopamine. In humans and other animals, dopamine is a naturally occurring chemical messenger in the brain. It regulates movement and sensation. With its dopamine knocked out, the roach cannot move freely or escape. Millions of years of evolution have refined this wasp's ability to hit her target and disrupt dopamine with the precision of microscopic brain surgery. This remarkable skill isn't learned, it's inherited, hardwired into each wasp. They are natural-born neurosurgeons.